Hello everyone. So when we last discussed core, we were talking about question 3F, which deals with non-disjunction, the lack of separation of chromosomes during meiosis, and specifically the non-disjunction of chromosome number 21 that leads to Down syndrome. Okay, so let's go ahead and restart where we were before we stopped class. So, question 3F. What we're talking about here is called a chromosomal aberration. This is not a birth defect. This is not a mutation. It's called an aberration. And what happens is this is when an offspring receives too many, or the opposite is also true, too few, or sometimes during crossing over, the pieces of the chromosomes aren't properly shared or aren't properly switched or traded. And so this can lead to broken or missing chromosomes. Anytime you have too much or too little chromosomal material, we refer to that as a chromosomal aberration. So, when we're talking about non-disjunction, this is a double negative. Because you'd think, well, if it's non and it's dis, these are both negatives, so why not just take that off and call it junction? Okay, well, there's a specific reason why. Because junction, this is where we joked about conjunction, junction, what's your function? A junction means to come together or to join. So what you actually have in meiosis is disjunction, which is to separate. This is actually a very, very good thing. In fact, it's required in meiosis for disjunction to occur. So you could actually say, we want disjunction. Disjunction is very important. It's very powerful. Well, if this is the typical condition, then you can then have non-disjunction. Whenever there's issues with biology, it's usually when the typical condition doesn't occur. That's disease. In this example, that's an aberration. It leads to an offspring that isn't typical. So if disjunction is to separate, then when this process doesn't occur properly, you have non-disjunction, which is to not separate. Don't forget, we're talking about meiosis here. And so in meiosis, you want to go from a diploid state to a haploid state. You want to go from 46 chromosomes in our species down to only 23. You want separation. You want segregation. So if you get non-disjunction, this is actually extremely harmful to the offspring. Now, what's interesting is it's harmful in animals. When plants get too many chromosomes, it actually leads to plants that grow more robust. They produce more fruit. And when you get into animals, if you have too much genetic material, if you have too many chromosomes, it's almost always life-threatening to the offspring. It almost always terminates the pregnancy. So when you're talking about non-disjunction in meiosis, non-disjunction in meiosis, we need to draw this out, of course. Okay, so let's get ourselves some circles, some cells. So what we're diagramming here is a very simplified version of meiosis that occurs in the ovaries of females or the testicles of males. And so you have 1 cell to 2, this is meiosis 1, and then you get a second division, and that's meiosis 2. Fine. So this is where we assign some colors. So we can go back up here And let's assign blue, and let's assign red. And if you don't have any crayons, don't worry about it, okay? Use pen and pencil. Use highlighter and pencil, okay? So blue, this could be what I received from my biological mother, whereas red is what I received from my biological father. Don't forget that we carry chromosomes in homologous pairs. So I carry 2 copies of number 21. One copy of number 21 I received from my biological mother, and the other copy of 21 I received from my biological father. So these are the chromosomes then that I am going to turn around, and I'm going to pass one of them on to Emily. Emily's either going to receive my biological mother's 21 or my biological father's 21. Now we focus on chromosome number 21 because of Down syndrome. It is by far the most common condition that's caused by a chromosomal aberration and also leads to a viable offspring. Usually, chromosomal aberrations lead to the death of the offspring or a spontaneous abortion. So there's many different things that could cause these chromosomes to not separate, but the classic example is that a spindle fiber breaks. Okay, so let's say that this spindle fiber breaks, right? So if that spindle fiber breaks, my biological father's chromosome, number 21, does not get pulled to the right-hand side. So what happens is when the plasma membrane pinches off, both of these chromosomes end up in this daughter cell of meiosis 1. So you end up with 2 copies of chromosome number 21 over here. And that's definitely not what we want. Like such. So since we attached the spindle fibers here and there was pulling, this would be anaphase 1. So then the chromosomes are pulled and both of them end up in this cell. Now, I'm not saying that there's no chromosomes over here, it's just that there's no number 21. Of course, there's 1 copy of number 1 and 1 copy of number 2 and 1 copy of number 3. There just isn't any copy of number 21. Well then, you get typical anaphase 2. There's not going to be any spindle fiber breakage here. And so now when these spindle fibers pull, you get 1 copy pulled to the left and 1 copy pulled to the right. 1 copy pulled to the left and 1 copy pulled to the right. Now what usually happens here is you only end up with 1 copy of each chromosome in each cell. But what you end up with here is 2 copies of chromosome 21. Now this looks like it should be the typical condition, but it's not. Because don't forget, this is the final daughter cell of meiosis. This is the product of meiosis 1 and meiosis 2. So these are actually gametes. So then if either of these 2 gametes are involved in fertilization, what you end up with is a gamete that offers 2 copies of chromosome number 21. That's usually not the case. Whereas over here, if either of these 2 gametes are involved in fertilization, they are missing a copy of chromosome 21. So let's diagram fertilization. We'll diagram fertilization using one of these gametes first, and then we'll diagram fertilization using one of these gametes. So here's the male's contribution to fertilization, the female's contribution to fertilization, and then here's your zygote. So here's let's blame this on the female. Let's say that the female has a non-disjunction event. Statistically, it is more common for females to have a non-disjunction event on chromosome number 21. We don't know why. The data is very clear that there's a correlation between mom's age and the frequency of non-disjunction for chromosome number 21, but we do not fully understand the biochemistry behind it. So here's the biological mother's contribution, here's the biological father's contribution, and here's their offspring. Here's the zygote. And now you start to see why it's so important that you had 2 copies of number 21 here. Because now, when you have fertilization, what the offspring ends up with is not 2, but 3 copies of chromosome 21. This is non-typical and is referred to as trisomy 21. Trisomy, tri because there's 3 copies of the chromosome, and then trisomy 21 because that identifies which chromosome the offspring has too many or too few of. This was first diagnosed by Dr. Down, and thus it is referred to as Down syndrome. Again, it is not a birth defect, it's not a mutation, it is a chromosomal aberration caused by the lack of separation of chromosomes in meiosis 1. All right, so if this was the gamete that was produced here, then let's see what happens with the gamete that's the opposite. Instead of having 2 copies being offered by the parent, let's see what happens when the parent doesn't offer any copy of a chromosome. So biological father, biological mother. So here, the biological father contributes 1 copy of 21. Now, the mother contributes 22 other chromosomes, just not any copies of 21. So here's no number 21. So now your zygote only ends up with 1 copy of chromosome 21. Well, instead of the first example being trisomy 21 because there's 3 copies of the chromosome, now you only have 1. So this is referred to as monosomy 21, or this offspring is monosomic. Now, you will never meet an individual that suffers from monosomy 21 because this is lethal to the offspring. This does not lead to a live birth and actually causes a spontaneous abortion and terminates the pregnancy. So there's a reason why I keep talking about the female and her contributing too many copies of chromosome number 21. The data is extremely clear here. IB likes to show you this graph. They either ask you to create the graph or they show it to you and ask you to explain it. 40, 50, and 60. So let's go for mom's age. The data set usually starts at the age of 18. It's much, much different to start collecting data on minors. That's a whole nother data collection. So these data sets typically start at the age of 18. So actually I should put the broken axis here. Should be a good role model. There's your broken axis. And this is mom's age. And again, I'll just keep stressing it. We know that this is directly correlated to the female's age, but the biochemistry remains unknown. Whereas the y-axis over here, this is frequency. This is the frequency of Down syndrome, or if you want to be clinical, trisomy 21. So if you're 18 years of age, the chance of you giving birth to a child with Down syndrome, it's about 1 in 2,000, which is actually pretty common when you get into the data of non-typical offspring. Once mom turns 30, the frequency doubles. frequency doubles into, it's only 1 out of 1,000. Actually, I drew that dot a little high. Once mom turns 40, here it becomes twice as common. Once mom turns 40, becomes 10 times as common, 1 in 100. Once mom reaches the age of 40, she has a 1% chance, 1 in 100, of giving birth to a child with Down syndrome. Once mom reaches 50, it becomes 10 times more common than at the age of 40. Let me line that up properly. Let's do that. And that's going to be 1 in 10. If mom is 50 years old, she has a 10% chance, 1 in 10, of giving birth to a child with Down syndrome. And so you get this exponential curve, like such. Okay? The most important point that you need to take care of this, or take from this, is why. We don't know why. The data is clear. It does not fall along any kind of socioeconomic line. It doesn't fall along nutrition. It doesn't fall along nationality. It doesn't fall by ethnicity. Regardless of mom's socioeconomic group, whether she's taken prenatal vitamins or not, whether she's had prenatal care or not, the one common factor between most Down syndrome offspring is that the age of their mother played a role in them ending up trisomy 21. Now, these individuals have a life expectancy of somewhere between 32 to 40 years old. They're always sterile. They have a broad shield-like chest, flattened nose, a broad forehead. They tend to have complications with their heart and have a whole host of respiratory difficulties. But they're also some of the most positive people you will ever want to meet. So this is non-disjunction of chromosome number 21 and leads to Down syndrome. And that would be core question 3F.